So welcome everyone. This is Teresa, and today I'm here with Professor Klaudia Brix to talk about research for our research catalog on this teamwork page. Welcome, Professor Brix. Please introduce yourself.
Yes. Hello, Teresa. Hello, everybody. So thank you very much for this initiative. I really appreciate that. So as you already heard, my name is Klaudia Brix. I'm obviously from Germany. And I joined Jacobs University. At that time, it was still IAB, about 20 years ago. Yeah, a long time. I teach in biochemistry and cell biology, but also in MCCB, so medicinal chemistry and chemical biology. And some of you also know me as the SPC of BCCB, and I'm your professor of cell biology.
Yes, thank you very much. What are you generally researching?
So in general, we work with protein trafficking in eukaryotic cells. So we work with cells and tissues and with a specific family of proteins. And these proteins are actually enzymes, proteolytic enzymes to be more precise. And they are called the cysteine, catepsin proteases, but also the kallikreins. So what we want to understand is really their role in health and disease. That means that we are using different models, in most cases actually cellular models, but we also used some animal models beforehand. And we try to understand the function of these proteases in, for instance, the thyroid gland, which is a very important endocrine organ that produces vital hormones. We also want to understand a bit more in detail glial cell and neural interactions, so a bit more like, let's say, hippocampal functions as well. And we are interested in the choroid plexus, which is a specific structure that is involved in the formation of cerebrospinal fluid. More recently, we got involved also a little bit into COVID-19 related research, because one of the enzymes we work with, namely catepsin L, is an enzyme that is able to prime the spike protein of the SARS coronavirus 2, and therefore it could, in principle, enable entry of the virus into cells. And since we found this enzyme on primary cilia, which are extensions from eukaryotic cells, we are also researching into that.
Yeah, that sounds very interesting. Thank you. What do you like most about doing research?
What I like most is that we have a complementarity of approaches that we can use, and that there's always something new that comes in. And I think that is staying curious in research and being open for new stuff. I think that is really what I find most interesting.
Mm-hmm. How can undergraduate students help out in your research team?
So the undergraduate students usually would start with, first of all, getting introduction into cell culture technologies, so to work with mammalian cells, and that sounds a lot easier than it actually is. So it really takes a while until the students are trained. Then we apply a lot of biochemical methods, which also are taught in the lab courses, for example, so protein purification, protein determination, and then separation, for example, by STS page and immunoblotting. These are like typical experiments that an undergraduate student would do. In addition, of course, in cell biology, we do a lot in imaging. So we either use antibodies or protease-targeted probes, so-called activity-based probes, that allow us to image the enzymes on the spot. And we use for that a very nice system, which is a confocal laser scanning microscope. And actually, the undergraduate students, I think, recognize our research group most for being involved in this imaging. However, another important task that is also becoming more and more popular is to not only produce nice pictures like the ones that you see in my background here, but actually to give meaning to them. So meaning to analyze these pictures and to provide numbers. And we would call this quantitative cell biology. And that would also be a task that the undergraduate students would perform.
All right, thank you. And finally, how and when can students join your research team?
So typically the students would ask me and approach me like after lectures or lab courses, for instance, or send me an email. and would notify me that they are interested in our research. And that is absolutely fine. And this will certainly happen in most cases in the 3rd or 4th semester, the latest. And then I think what you also know from the previous years and the current BCCB students is you have to work hard. So in principle, it might sound just nice to join a research group, but if you would join, for example, my lab meetings, I would also ask you to prepare a presentation or to summarize some scientific content. So it is not only just sitting there and listening, but also probably then getting active.
All right. Thank you very much. I think everything sounds really interesting, and I hope a lot of you can join Professor Brix's research group in the future. Thank you very much for the interview.
Thank you very much, Teresa. I should also add, perhaps at the end, that we are a rather small research group. So do not expect that, too many students can actually join. But I'm, of course, welcoming everybody to the lab meetings, and then we can discuss further. Thank you very much.
All right. Thank you.